Now, take out your pencils. You'll have to go the expiratory system and learn the parts of the functions. Yes, there's a lot of noise. Now all of you start drawing the diagram. All of you take out your pencils. Okay. Done. Take out your books now. Stop talking. When you draw the expiratory system, remember again the position of the kidneys when you draw. The position of the kidneys when you draw. Remember to draw the right kidney little bit lower. The right kidney will be little bit lower. Remember when you draw the diagram. Because of the position of the liver. The liver is on the right side, that's why. Surgery. Surgery में पूरा किडनी नहीं निकालते. Even if it is a cancerous situation, तो पूरा किडनी नहीं निकालते. It's part of the. Start. Just 10 minutes for the diagram. When I am drawing the diagram, you are taking the privilege of talking because मैम तो वैसे भी इस तरह की देख रही है. so much to talk. इतना क्या है शेयर करना, क्या इतना क्या बोलना है? हाफ एन आवर भी अगर ब्रेक तुम लोग को दिया जाए तो भी वो कम ही होगा. कम है ना? Talk non-stop. आज कोई चिप्स देने नहीं गया? क्यों? किसने पूछा क्यों? मम्मी ने ब्रेकफास्ट दिया ना, उसने नहीं दिया. Start. Without talking, if you draw the diagram, you'll be able to understand something. This is the right kidney or the left kidney? Left kidney. diagram should be bigger than this. Diagram has to be prominent, clear. Paths should be not very clumsy when you draw the diagram. So always I have mentioned your diagram should be minimum when you are drawing in your rule book. Your diagram should not be less than 10 to 12 lines, and these are big diagrams. If you draw the stomata, that is still understood 5 to 7 lines, you can finish up. But the diagram of the heart and the excretory system and the respiratory system and the digestive system, you cannot finish up in 5-6 lines. The path should be neat, well shown, labeled correctly and properly. When you are drawing it small, everything is becoming so congested and the parts are not understood properly. Always draw a neat diagram. So diagram drawing, see, hardly in your entire portion you have got just 10-12 diagrams only. So those diagrams, just few times you practice, easily you can pick up those 2-3 marks. So easy. You don't have to understand anything there. You don't have to learn and remember anything there. Just practice few diagrams. You know the labels. Little bit neat, little bit as per the shape in the book. Easy to pick up those 3 marks. Finish up. What happened? Huh? What are you talking about? Sphincter muscles. S-P-H-I-N-C. S-P-H-I-N-C-T-E-R. Sphincter muscles. Renal artery, renal vein. Done? All of you had finished, no? The last 2 diagrams that I asked you to draw, digestive system and respiratory system. Please practice those diagrams. Diagram of the heart, structure of the heart. Those of you did not finish the other day, you should finish it up. Maybe the next class or something, I may again check your book. So by that time, you should be done with your work. Life process in which of your which schools it is already over? Konsa? And already over. And did you start with any other chapter after this? No. The next class I'm going to take a test on this chapter. किसने बोला ये? बोला किसने? इसने बोला? नहीं मैंने कुछ Today we had actually only talked about, for you, we had only talked about transportation in plants today. Till the last class we had already finished some major parts of the chapter. So, how much of the chapter is done in your school? Both the answer you said. How much of the chapter is done in your school? मैम वो चलेगा और 1 आध महीना हमारा different sections ना बोल सकते हैं तो पहले तुम्हारा कंट्रोल कोऑर्डिनेशन टेस्ट कर ले बोलूँगा तो वो सही है तो जाके होगा respiration in plants Respiratory system, you mean? Respiratory system is going on. Acha. Now, what I'm asking you is, if I take a test the next day, will you be able to write answers? किसने बोला नहीं? I'm asking only her because she's a new admission. You don't dare to say no. Shameless creatures all sitting here. The question is only for her, not for you all. Question, what's your name? Prisha. So Prisha, you had missed all this, the main three parts of the chapter. So अगर मैंने test लिया next day on if I take a test next day and I will take a test basic concepts, I'm not asking you to learn, go through and memorize the entire day. Basic concepts समझो of nutrition, respiration, excretion चलो I am not taking the major three parts of the chapter. Will you be comfortable to write the answers? You go through the parts जो आता है, because after that if you want those topics to be taught, you'll have to talk to Pramila and she'll help you how to sort out. But then you go through the chapter, see how much you are able to understand the notes and the questions you'll take down from your friends, whatever question answers I have discussed with them, whatever notes I have given them, that you take down from your friend. Okay. Now, let us start with all of you are done with the figure. Nutrition, respiration, transportation, basic concept, जो question answers किया है, volume के questions go through. I will give you a test from the questions of the volume only. तो volume के जितने भी questions हैं, go through all. Basic concepts आने ही चाहिए. If I am asking you to learn and the entire process of double circulation and write, of course that needs little bit of study and practice. You have to practice the points and remember, right? But I will be taking a test only on the basic concepts, the major definitions and the major differences and the term, the names, only that. So that much you should be able to remember. No diagrams now, later. Now look into this figure. We are talking about the excretory system in human. The excretory system in human. Now, what are the major parts of the excretory system and where is this system present in our body? To talk about where is this excretory system present in our body? It is present in the dorsal side of our body. Now, which is the dorsal side of our body? To understand the dorsal side of the body is this side. The dorsal side of the body, where just above the hip. Where is this present? Just above the hip in the dorsal side of the body. How are the kidney placed? The kidneys are placed on either side of the backbone. So when we are talking about the location of this entire system is on the dorsal side of the body. The kidneys are always placed on either side of the backbone. And remember, this is not in the front of our body. This is the ventral side of our body. I am constantly mentioning it is on the dorsal side. I asked you to remember when you draw the diagram to draw the right kidney little bit lower. Why do we draw the right kidney little bit lower? The right kidney is little bit lower because it is pressed by the liver. The liver is also on the right side of our body. The liver is a big organ that we have on the right side of the body, and because of the position of the liver, on the right side, the kidney is little bit pressed. So now the right kidney, that's why a little bit lower than the left kidney. They are not exactly at the same level when you are drawing also to remember. So the excretory system, what are the important parts now? What are the important parts of the excretory system? Here we have drawn all of the organs along with the blood vessels, but which are the most important parts? The excretory system consists of a pair of kidneys. The excretory system consists of a pair of kidneys. What next? A pair of tube-like structure known as ureters. A single urinary bladder, and fourth is a single tube-like structure known as urethra. So what are the parts of the excretory system? These are the four main parts of the excretory system. We talk about the parts of the excretory system as the kidney and the ureter, the urinary bladder, and the urethra. So these are the four parts. But apart from these four parts, we have also drawn the blood vessels. What is the relation of the aorta? I'm drawing the aorta with reference to the heart. What is the aorta doing? Carrying oxygenated blood. Today also we were discussing that aorta is the major artery of the body which carries oxygenated blood from which chamber?
Left ventricle.
Left ventricle. From the left ventricle. So now, today also we were discussing that the left ventricle should be contracting at a great force so that it can throw the blood into the aorta. And from the aorta, a single branch now that we are talking about is what? The renal artery which will carry the blood towards the kidney. Such type of arteries coming out from the aorta, developing from the aorta are many, supplying blood to other different parts of the body also. Right now we are only talking about the renal artery. So now that I talk about the renal artery, what type of blood then the renal artery is carrying towards the kidney? Oxygenated. I have drawn in blue and red also. So now the aorta is carrying oxygenated blood. One such branch of the aorta is the renal artery, and the word renal, the moment I talk about, renal is always to do with the kidneys. Renal artery, renal capillaries, renal vein, always understand this is something related with the kidneys. So when we are talking about the renal artery, apart from the main excretory organs, I first of all speak about the blood vessels. The aorta carry oxygenated blood. The oxygenated blood is moving towards the kidneys. And we have already understood what is the function of the kidney? Filtration of the blood. But if the blood is already oxygenated, why the need for filtration? Because we are talking about oxygenated blood, but the blood is already containing a whole lot of waste substances. What type of substances majorly to understand? The nitrogenous waste substances. Those nitrogenous waste substances are to be thrown out. Those nitrogenous waste substances are to be filtered here. Remember, the blood which is carried by the renal artery is already having a lot of oxygen now. Here with reference to the kidneys, we are not talking about the purification or the filtration reference to oxygen and carbon dioxide, but we are only talking about filtration with reference to the nitrogenous waste. Understanding? And now, the blood which is coming out. So if I have to show here what type of blood is entering the renal artery and what type of blood is coming out from the renal vein, obviously the blood which is coming out from the renal vein, coming out from an organ, the blood which is coming out from an organ is always deoxygenated. But now with reference to kidney, I will say the blood which is entering the kidney is having a very high concentration of nitrogenous wastes. What are the prominent nitrogenous wastes I mentioned already? Uric acid. Okay, urea, uric acid. For the time being, always remember urea, uric acid. Two major nitrogenous waste substances. So which blood is supposed to have a high concentration of urea, uric acid? This blood or this blood? The oxygenated blood. And this is getting filtered here. So the blood here, which is deoxygenated but has a lower concentration of urea, uric acid. Understood this concept first? So now we try understanding again what is the job of the kidney and the ureter and the urinary bladder, some of the special features to understand. So when we are talking about the kidney, what are some special features of the kidney? What is the shape of the kidney? The shape of the kidney always to write. It is bean-shaped. It's a bean-shaped reddish organ. So when we are talking about the kidneys, it is a bean-shaped reddish organ. What is the location? It is placed on either side of vertebral column. What is the location? Placed on either side of the vertebral column. Placed on either side of the vertebral column. What is other word for the vertebral column? Other word for the vertebral column. Vertebral column को commonly what is it? नहीं, backbone. Backbone के अंदर जो nerves है उसको spinal cord बोलेंगे. The bony casing is not spinal cord. The bony casing is backbone. Remember. So we are talking about the bean-shaped structures. Position क्या है on either side of the backbone, vertebral column. What is when we are talking about this kidney? The kidney has the section of the kidney. If you try to see, the kidney has two specific regions. If you talk about the section of the kidney, the outer region of the kidney is known as the cortex. अच्छा नहीं लग रहा है? The outer region of the kidney is known as the cortex, and the inner region, which is lighter, that is known as the medulla. Medulla. So now, when we are talking about the kidney, say suppose I talk about the section of the kidney. How would the section of the kidney look like? The outer region, I'm saying, the outer region is darker. What is this outer darker region known as cortex? And what is this inner region known as medulla? The outer region cortex and the inner region known as medulla. So longitudinally, when we try to look into the section of the kidney, it has got many more parts inside, but only to remember, the outer darker region is known as cortex and the inner region is known as the medulla. Here, when we talk about that, what is actually present in this entire region? In this entire region, a large number of tube-like structures, something like this. Some tube-like structures are present, which finish up this way. Some tube-like structures are present, running down from the cortex to the medulla, finish up in the cortex. Tube-like structures running down from the cortex to the medulla, finish up like this in the cortex. Such type of tubular structures are in millions in the kidney. Such type of tubular structures running from the cortex to the medulla and coming up here. Those internal structures you don't need. I just give you an idea that what is the basic concept of the structure, the cortex and the medulla. You are only to learn this one. That right from the cortex down till the medulla and back in the cortex, if you try understanding, there are a whole lot of some tubular structures. And what are those tubular structures known as? These tubular structures are known as the nephrons. Those tubular structures are known as nephrons. What are these? The kidney has millions of tubular structures known as nephrons. And how will we define these nephrons? Nephrons are the structural and the functional unit of the kidneys. So what is the kidney made up of? The kidney has got the cortex and the medulla, and look into the cortex and the medulla in detail. We come across that there are millions of such type of tubular structures. They are performing the function of the filtration of the blood. And what will we name this as? Underline, write this word and write on the definition also. What are these? Nephrons. Definition same like the alveolus. What was the definition of alveolus? Structural and functional unit of kidney. Lungs, same that way, remember? We are talking about the nephrons now. Where are nephrons present? Inside the kidneys. What are they doing? Filtration of the blood. Write down. Nephrons, structural and functional unit of the kidneys. What are nephrons? Or name the structural and functional unit of the kidneys, nephrons. So what are nephrons? The structural and the functional unit of the kidneys. Write down the definition. So when we are talking about the kidneys, the first feature is that the kidneys are bean-shaped, reddish structures, and then the kidney consists of millions of such type of tubular structures, the nephrons. We'll talk about the structure of the nephrons later. This kidney gives rise to some tubular structures like this, and the broad end of the kidney, which gives rise to the tubular structure known as the ureter. This is known as the renal pelvis. So talking about some additional parts of the kidney, I'm saying that this kidney, I already mentioned that it is a bean-shaped structure, and also to remember that the outer wall is convex. It is a convex curved structure there, and it has got the medulla. The medulla always gives rise to a broad. The medulla always gives rise to a broad structure, and this broad structure is known as the pelvis. Gives rise to a broad structure, and what is this broad structure known as? This broad structure is known as the pelvis, and from this pelvis a narrow tube would emerge out, and this is what we are talking about as the ureter. So basic about the kidney, what are some of the points that you are supposed to remember? What is its location? What is its basic shape? And what is it made up of? Now, this kidney has got a broad opening. The medulla of the kidney has got a broad opening. What is this broad opening known as? I already mentioned this part is known as the renal pelvis, or say only the pelvis. So that is about the kidney. Now I talk about the ureter. What are ureters? Ureters are narrow tubes. What are these ureters? Ureters are narrow tubes. What are these narrow tubes doing? What would be their function? Passage of the urine. That is their function that you are supposed to write down. What is the function of the kidney? I already mentioned. What is the function of the kidney? Filtration of the blood, maintenance of water balance, salt and water balance. So the same thing that you will write there also. So after this, when I am talking about that, what is this ureter doing? The ureter is carrying out the waste substances after the filtration of blood that is getting accumulated in the kidney. So what is the job of the ureter? Take the waste substances and pour them into the urinary bladder. What is this ureter made up of? The ureter is made up of the smooth muscles. It is made up of smooth muscles. It can undergo the process of contraction, relaxation as per the need of the body, involuntary muscles. So that is about the ureter. Ureter is connecting which parts to which part? The kidney to the bladder. What is its job? Carrying the waste substances from the kidney to the bladder. What is the substance it is made up of? What tissue is it made up of? Smooth muscles. Talk about the bladder now. When I talk about the urinary bladder is also made up of smooth muscles. What is its function? It stores the urine for a particular period of time. It stores the urine for a particular period of time, and under nervous impulse, only under nervous impulse, the sphincter muscles will contract. It is only under the nervous impulse. Now, what is the meaning of nervous impulse? Under the message from the brain. Only under the message from the brain, these sphincter muscles, which are actually at the junction of the bladder and the urethra. Junction of the bladder and the urethra. Ureter, and this is urethra. So what is about the urinary bladder to remember? It is made up of smooth muscles. It is storing the urine for a short period of time. And under the nervous impulse, the contraction of sphincter muscles takes place. The contraction of the sphincter muscles will take place. And these sphincter muscles will allow the flow of urine. From where to where? Flow of urine from the bladder to the urethra and out of the body. So if I now have to talk about the urethra, then what is the job of the urethra? The type of the tissue that the urethra is made up of, the bladder is made up of, and the ureter is all the same. The ureter, bladder, urethra, all made up of the same type of smooth muscles. The property, involuntary function, almost the same. But especially only when I talk about the junction of the bladder and the urethra, that is regulated by the nervous impulse I already mentioned. And if I have to mention that what is the job, this is what a difference you are asked also, the ureter and the urethra. So if I talk about the urethra, then what are you supposed to write? Releases the urine from the bladder to outside, to the outside of the body. Remember, when we write down the function of these two parts, very, very important, you should know how to differentiate them. It seems very easy, but when you are writing them, you may not get complete correct answers. What is the function of ureter? Passage of urine from the kidney to the bladder. The ureter does not help the release of the waste substances outside. So which is that tube which is allowing the release of the waste substances outside from the bladder? Urethra is that tube. Urethra. So the urethra will always help in the release of such type of structures, such type of waste substances away from the body. But from time to time, the sphincter muscles will contract, just like the valves in the heart or the veins. This will contract and they will become smaller, make the pathway for the release of the urine. So when will the nervous impulse be generated? The nervous impulse will be generated only when the bladder is partially full. The bladder partially full, then the brain sends the message. There is a particular region in our brain, you will learn in the next chapter. There's a particular region in the brain known as hypothalamus. There's a particular region in the brain known as the hypothalamus, and the hypothalamus keeps on giving messages to different parts of the body. The nervous impulse that we talk about as impulse means basic information, remember. We'll talk in detail in the next chapter. Excretory system, only this much you'll have to understand. The basic parts, what are the parts now of the excretory system? A pair of the kidneys, pair of ureter, a single bladder, single urethra.
I have a doubt that vena cava will take the blood from kidney to the heart, right? So the arrow will be up.
Vena cava will take the blood. I'm sorry, I made a mistake here. So this vena cava will have. Absolutely is correct. So the blood, I explained this, but the arrow is wrong. All of you check this. Correct. So the arrows, when you talk about the arrows, the aorta will carry the blood by the way of the artery to the kidneys.
So that will be the oxygenated, right?
That is oxygen. I explained also, no. Now the blood which is entering the kidney is oxygenated. Sometimes you are asked, the blood entering the kidney is oxygenated, then what is the need for filtration? Filtration of what is happening there? Filtration of nitrogenous waste. So that word you have to mention in your answer. Filtration of nitrogenous waste. Kidney has nothing to do with separation of oxygen, carbon dioxide. वो kidney का job नहीं है. Then what is the kidney doing? Only the separation and throwing out of the nitrogenous waste. Okay. So structure, this is what you have to study. Kidneys, what is the location, shape, structure, function. Ureter, bladder, urethra, what are they made up of? What is their basic function? Now if I ask you to write one question there. Write one difference between the ureter and the urethra. One difference between ureter and हर बार कुछ ना कुछ दिएगा. Write one difference between ureter and urethra. तुम लोगों ने उसको भगा क्यों दिया वहां से? One difference between ureter and urethra. What did I tell you? What should you write? अच्छा, structure क्या लिखेंगे? Structure if you have to write connects which part to which part and the function. So you may be asked anything. Structure पे difference लिखना है, function पे difference लिखना है. So write down the first point. Ureter connects the kidney to the bladder. Ureter connects kidney to the bladder. Ureter connects kidney to the bladder. Other side. Urethra. Urethra opens the bladder to outside of the body. Opens the bladder to outside of the body. Or to the outside of the body. So it is an opening of the bladder. Second, now the function. Ureter carries urine from bladder, from the kidney to the bladder. Carries urine from kidney to the bladder. And urethra releases the urine out of the body. Releases the urine out of the body. Now, next we try to understand that I already showed you that talk about the cortex and the medulla, what is present inside the kidney now. We try understanding the parts of the system is already over. Now we try understanding what is there inside the kidney. Inside the kidney there are billions of such type of structures. What are these structures known as nephrons? What are these nephrons doing? Filtration of the blood. How are these structures majorly? With as if a knob-like structure, a head-like structure, and the entire long tubule. So what are these structures called? These structures are called the nephrons, and the structure would be something like this. These structures are called the nephrons, and the structures are something like this. We are going to draw this structure, understand the parts of the functions. So now that we talk about the nephrons, extensively coiled tubular structure. So remember, I have been drawing that head-like structure. The head is this part. The head-like structure that I draw is this part that we are talking about. In this head-like structure, when we magnify and see, it is a cup-shaped structure. एक cup shaped structure होता है जिसको Bowman's capsule कहा जाता है. This is known as the Bowman's capsule, and this Bowman's capsule will have a knot of blood capillaries. एक bundle of blood capillaries होता है इसके अंदर. So there's a bundle of blood capillaries inside, a network of blood capillaries inside, जो renal artery का division से ही बना है. Renal artery. So this renal artery, as it enters, it again divides into a large number of capillaries, large number of vessels, and ultimately those vessels result in the formation of a ball of blood vessels. So this ball of blood vessels is what we are talking about as the glomerulus. Glomerulus. From this glomerulus, again, a network of tubules will come out and wrapping around the entire part of the nephron. But talking about the part of the nephron, what is this entire structure? I'm talking about nephron. How did we define the word nephron? Of the kidneys. So ये हो गया Bowman's capsule, glomerulus. Next what? The entire tubular region of the nephron. The tubular region of the nephron is the longest part of the nephron. The entire tubule is divided into three major parts based on their function also. And then ultimately the collecting duct. See that the tubule that I've drawn also with a head-like structure, then some tubule running from the cortex to the medulla and फिर से वापस cortex में जाके they wind up. So this entire tubule that we get to see and surrounding the blood capillaries, this is what is responsible for the process of filtration of the blood. Now, filtration of the blood kaise? We are going to learn all the steps and the processes. As the blood goes here, some amount of filtration takes place here, followed by the entire region here. If you see the entire tubular part, what is there? Surrounding the tubular part, there's a whole lot of blood capillaries and a whole lot of diffusion takes place. Exchange of substances taking place, and that is how the filtration of the blood happens. Ultimately, the waste substances all get drained up here. इसको क्या बोलेंगे? Collecting duct. So अब collecting duct के बाद, from the collecting duct, the waste will be thrown here, and then the rest follows. We are going to learn now how exactly the parts of the nephron and how the process of filtration takes place here. To understand what are the parts of the nephron, first of all, get acquainted with the names. What are the names? What are the parts of the nephron? First of all, we are talking about the structure like this. What would this vessel be? This vessel that I am drawing is which was that vessel that we had drawn, which is entering the kidney. We were talking about the dorsal aorta. Now we are not talking about the dorsal aorta. Now we start from the renal artery. So this is the renal artery. The renal artery, as it enters into the knob-shaped structure, this renal artery divides into a large number of capillaries. The renal artery divides into a large number of capillaries, and these capillaries now, they again results in the formation of a network of tubules again. So first of all, what am I drawing? What is this particular vessel? This particular vessel is the renal artery. What type of blood the renal artery carries, you are already aware. Now, this renal artery is divided into another set of vessels. What are these set of vessels known as? These set of vessels are known as the broader vessel which is moving towards the nephron. This is known as afferent arteriole. Get acquainted with the words. These are known as afferent arteriole. We will draw. You first understand. So this is the afferent arteriole. The afferent arteriole, how is the afferent arteriole formed? The afferent arteriole is formed by the division of the renal artery. The afferent arteriole is formed by the division of the renal artery. Renal artery again divides into a network of blood capillaries. What is this network of blood capillary? Known as glomerulus. What is this called? This is known as glomerulus. How will you define the word glomerulus? Glomerulus is a network of blood capillaries formed by the division of the afferent arteriole. Is the definition correct? Now check. What did I say? ये है renal artery. Renal artery divide होके a number of blood capillaries. First of all, the afferent arteriole. Afferent arteriole divide होके a number of blood capillaries, extremely fine, thin, narrow capillaries. And what is this network of blood capillaries known as? This network of blood capillaries is known as glomerulus. But glomerulus is surrounded by a double-walled cup-shaped structure like this. ये double-walled cup-shaped structure को क्या बोलेंगे? This double-walled cup-shaped structure is known as the Bowman's capsule. What is it? The Bowman's capsule. अब ये glomerulus और Bowman's capsule together, which forms the head part of the nephron, इसको बोलेंगे Malpighian body और Malpighian corpuscle. Why worried हो गया ना ये चैप्टर स्कूल में? तो ये सब है सिलेबस में, कैसे नहीं पढ़ाया? एक्सप्रेशन नहीं हुआ? हाँ? डिटेल में नहीं पढ़ाया तो नेफ्रॉन नहीं पढ़ोगे तो कैसे होगा? ये डायग्राम आएगा एग्जाम में?
सिर्फ मोमेंट पढ़ा के छोड़ दिया। हाँ।
उसके बाद आगे का नहीं
हेनली लूप
corpuscle was there and glomerulus was there. Other than that,
तो प्रोसेस कैसे समझाया?
वो हमारे
not that much details.
Nephron diagram you get in the exam to draw. Diagram of the nephron you will get in the exam to draw. इसलिए I am drawing in parts. You will not draw the diagram now. You understand first. So ये हो गया the first part of the nephron. That is the head part of the nephron that I had drawn. तो head part of the nephron में क्या क्या parts है? Glomerulus and Bowman's capsule. Bowman's capsule क्या है? It's a cup-shaped structure, double-walled cup-shaped structure. अब देखो inner wall जो मैंने draw किया है, inner wall I have drawn dotted. Now this is only to make you understand. ये जो inner wall है, इसमें ना pores रहते हैं, openings रहता है, which also help in the process of filtration. And the outer wall is thick, even. Clear? So I'm talking about the renal artery जो divide होके afferent arteriole बना. Afferent arteriole divide to form another set of capillaries. This set of capillaries known as glomerulus. Glomerulus is put as if in a cup-shaped structure. वो cup-shaped structure को बोलेंगे Bowman's capsule. ये both these parts together known as the Malpighian corpuscle or Malpighian body. ये डायग्राम बुक में है. तुम्हारा बाइबल देखो. हाँ. तो वो एक ही book, the same book that we read. No change in the syllabus. So हम लोगों ने पढ़ा in our time से अभी भी तुम लोग वही पढ़ रहे हो. हाँ. This diagram is there in your syllabus. ये practice करना पड़ेगा my sweethearts. स्कूल में पढ़ाया नहीं बोल के don't be so cool and casual कि एग्जाम में आएगा नहीं. It comes in the paper. Diagram of the nephron, it comes in the paper. So now Malpighian body. Just look at it. So body weight कितना होगा? About 20 kg.
42.
11 12 standard तो बुक कैसे उठाओगे तुम?
और इसकी टेंशन आप कर रहे
1
हो
2 3. This Darshan also, just look at it.
यार दर्शन का मेरे से मैम strength also matters मैम, you can't judge a person by its अच्छा
Strength है.
हाँ है
He did not like it. Are you okay? What happened to you? Okay, now when we are talking about this part, when we are okay, take it easy. Take it easy. Now, when we are talking about the parts, again, when we talk about the parts, what is Malpighian corpuscle? Malpighian corpuscle is made up of this. So this is the first part. Now we talk about the tubular part, which is extremely long. Now we talk about the tubular part. How about the tubular part of the nephron? The tubular part of the nephron is extremely coiled. When we talk about the tubular part of the nephron, it is extremely coiled. So we talk about the major parts of the tubule. Let us understand that the first part of the tubule would be more or less present in the cortex. We talk about the tubule more or less present in the cortex. Then we are talking about a very, very long U-shaped structure, and this is known as the Hanley's loop. So this is known as the loop of Hanley. We are going to label all these parts. So we talk about, first of all, extremely long shaped tube known as the Henle's loop, and then this is followed by the distal region of the tubule. So we are going to complete and followed by the collecting duct this way. Now let us look into the other parts and label them. So this would be the major structure of the nephron. So when we are looking into the entire structure of the nephron, is even more coiled. But these are the parts that you are going to draw. How will you draw this part of the nephron? This is the first part. This is the first part known as the proximal tubule, the first part of the nephron. Now in your book, they have mentioned as the first part of the nephron. So this is a part of the tubular region. So I would name it as proximal. Now this names bit, I can say that this is optional for you. If you can remember the name, you write. Or else you can just write down the tubular part of the nephron also, it is okay. What is this known as? This is known as the proximal convoluted tubule. Proximal convoluted tubule. What about the second part? This is known as the loop of Hanley. Proximal convoluted tubule. Correct? The proximal. Why the word proximal? Because it is close to the proximity. What is the meaning of proximity? Close. Close to the renal corpuscle. That's why. Then we talk about the loop of Hanley. And what is this particular tube known as? This particular tube is known as the distal convoluted tubule. What is the meaning of the word convoluted? Extremely coiled. And the last part is known as the collecting duct. What is this last part known as? Collecting duct. So what will we name this as? Collecting duct. So what are the parts of the nephron that we have drawn? The parts of the nephron first of all are this. Let us complete now. We are talking about the blood vessel, the renal artery entering by the way of the afferent arteriole. Afferent arteriole dividing again to form a network of capillaries known as glomerulus. Glomerulus is present in a cup-shaped structure known as. Together they form what? The Malpighian corpuscle or the Malpighian body. So that is okay. Now we talk about the tubular region. What are the three parts of the tubular region? 1, 2 and 3. In your book they have just mentioned as tubular part. But when you describe the process, it's always better when you know the names. Otherwise, describing the process becomes difficult for you also to remember. So what are the three major parts of the tubule? The three major parts. This is the proximal. This is the loop of Hanley and this is the distal. Distal means distant apart from the Malpighian corpus. So that's the word distal. So proximal, distal, and between a very long U-shaped tube known as the loop of Hanley. Correct? And lastly, we talk about the collecting duct. What about the blood vessels? Let us complete with the blood vessels also. Now, this blood vessel, which is formed again by the capillaries, by the union of the capillaries, again they are formed, and they are now coming out, emerging out from the nephron. What will we call this as? This is known as the efferent arteriole. One is afferent, the other is efferent. Can you tell me a difference when I draw the diagram? What is the difference you are able to understand from the diagram?
Yeah.
Huh?
One is going inside.
Afferent is going outside. Afferent is coming inside. Afferent.
एक और difference.
Ma'am, afferent arteriole is dividing into multiple. Coiled.
एक और difference है efferent or efferent. The way I draw, कुछ भी ऑडिट में तो नहीं दिया है. That is narrow. Always to draw also when you are drawing the diagram. Efferent is broader. Efferent is broader, and that is also one big reason why the filtration happens here. So when you draw the diagram, the efferent always to draw broad and efferent always to draw narrow. We learn what is the difference in the circulation. So now when we are talking about the entire efferent arteriole, now divides into a network of capillaries, and this network of capillaries are all connecting, all surrounding the entire tubular part. You don't have to draw double line capillaries for this, only single line capillaries for you is enough. And then these capillaries will again unite and result in the formation of another set of tubes, and this tube is what we are talking about as the renal vein. So this is the complete diagram that you'll have to draw. I'll give you 5 minutes to draw the diagram now. No? I have drawn in parts. Chalo, try karo. Understand and draw the diagram. If you understand and draw, it's very easy. Okay, chalo, fatafat karo. Look, this way. You will draw the diagram this way. Don't draw horizontal. You will draw the diagram this way. Clear? Don't make your diagram too broad. जहाँ पे fit हो जाना चाहिए diagram. Make it narrow. Don't make it so broad. करो fast. We'll understand the process also today. This entire part should fit in almost इतने से space में होना चाहिए. Start drawing as I have started. इतने से space में तुम्हारा diagram हो जाना चाहिए. Don't make it so broad and spread out. Don't make your diagram broad and spread out. हाँ, parts to label करोगे. Now for this diagram, since you have to label some parts on the right and the left also, you may draw the diagram in the middle, in the center you draw the diagram. एकदम ऊपर से नहीं draw करो. Whenever you are drawing a diagram, most of you have a habit. एकदम ऊपर से start करते हो. And then everything becomes so clumsy at the top. थोड़ा सा नीचे से start करो. What happened? यहाँ से start करो. Then you get space here. You also. कहाँ से भी space मिलेगा नीचे? Start your diagrams little bit from below the margin. Don't start exactly from up there. You have to label the parts. Afferent arteriole, efferent arteriole. वो सब label करना है. You will not get enough space up there. Start. Fast, fast, fast. हो गया?
हमको तो दिखा है. हम बहुत अच्छा बना है.
दिखाओ.
अच्छा बना है इसलिए तो बोल रहे हैं.
ये डायग्राम थोड़ा इलाबोरेट है. इतना छोटा सा स्पेस में कैसे बनाया? You make this part first. All of you make this part first. पहले ये part बनाओ, then you make the blood vessels, the tubule and this. First you make this, those lines that I've drawn in black. First you make this, then you make the blood vessels. Then you understand how to draw the diagram. ये जो मेन बॉडी ऑफ द नेफ्रॉन जो मैंने ब्लैक लाइंस में बनाया है ये पहले ड्रॉ करो, अदरवाइज पता चलेगा बाद में when you draw the blood vessels and the nephron they will be almost of the same length, वो अच्छा नहीं लगेगा. No, what I told you, draw the part of the nephron first. Draw the part of the nephron first. Always you should take the diagram this way. This part should be drawn first. ये हो गया. फिर अभी तुम ये करो. इसको कर्व करो. 2-3 कर्व देके. So ये हो गया. Now you draw the renal artery. Understood? ये ट्यूब छोटा होना चाहिए ये सारे ट्यूब से. You will draw the diagram shading. When you draw, see now, for me I can use 2 colors. You cannot use 2 colors, then how will you differentiate the tubes and the blood vessels? The tube of the nephron should be drawn a little bit dark and the blood vessel lighter. So that's how you'll be able to differentiate. Narrow hoga. This will be much narrower than this. Then you complete the tubule first. Then ये blood vessels थोड़ा सा light draw करो. These lines that I draw in black, you draw it bit bold, dark. Blood vessels, the ones that I have drawn in red, that you should draw a little bit lighter, but prominent. Then your diagram will look neat and nice. Good. This is good. Good. Blood vessels? Good. This is a difficult diagram, but at least if you are able to draw it of a proper shape, you will easily get. Show, keep showing me what diagrams you are drawing. Yes. Draw the tubule first and then the blood vessels. Pen you use color pencils also you're not allowed to draw use. Own अभी के लिए किया it's okay, but in the exam you're not allowed. Only pencil. So how will you differentiate? See, if you keep drawing the diagram only with pencil the same shape, these differences you will not be able to see. Everything looks so clumsy in the diagram. So to make it prominent, you will have to draw this black lines bit dark and the red lines bit lighter in shade. Sure. Make it fast. How many diagrams so far we have drawn? बहुत सारा. Number बताओ.
5.
All those diagrams that I have taken up in the class and I have asked you to draw, these are all important diagrams you'll have to practice. Number 1, stomata. Number 2, nutrition in amoeba. Then digestive system, respiratory system, structure of the heart, excretory system in human, and the structure of the nephron. For life processes, only these few diagrams to practice. Make it fast. Sometimes with a question also explain with a diagram, explain with a well-labeled diagram of the respiratory system, the process of gaseous exchange. So you should be able to draw the diagram of the respiratory system, otherwise that 2 marks as it is gone. These lines you don't, no need of giving double lines and make it prominent. Just single line to give is enough for you. For 2 to 3 marks, this is enough and enough. Very good. Just make it make it straight. Start labeling the parts. The last line. Constantly you are talking. Are you done?
Yes, ma'am.
और बाकी
ये थोड़ा लेवलिंग करना है ये तो बैठ के गाड़ी ऐसे जाए
भाई रुक जा मेरा बनेगा मेरा बनेगा तब तो सीखी
already 10 minutes over. See the glomerulus part, make it bit prominent. The inner wall of the Bowman's capsule, thora prominent draw karo, and on inside the glomerulus prominent draw karo. Show your diagram. This is what I am asking you. See here. This is how you draw. Roshi has used red pen, not to use red pen, but then you can make this blood vessels light like this with the pencil itself, and this part you can use black pen, or make the diagram darker with the pencil itself. That you can do, but don't use color. This way, neat diagram you try to draw. Show others.
नहीं पता ना
This part is okay. This part is good. Make the blood vessels fast. Henley. Henle. Good. Good. That's also good. But you don't use colors. You can use black pen instead. Now label the parts. Once you understand the diagram, is it not easy?
yes
Easy? Understand the diagram. That which of the parts I have to draw. You can use black pen, but no other colors, remember. Labelings, plan your labeling so that you don't make it clumsy. This, and then this one. The tubular part, 3 parts. And then the collecting duct. Make it fast. Finish up now, label. Good. Label. There, these last 2. Show me your diagram. Label all the parts. Oh, that this chapter is over? No, what you said, till which part of the chapter? Nutrition only.
No, it's that. It's not that.
Nutrition. Okay, now those of you did not finish, you will complete this diagram at home. You've taken a lot of time now. Done? This side. Now, with this structure of the nephron, we have to understand the next process. What is this? The process of filtration of the blood. How does the filtration of the blood take place? How does the filtration of the blood take place? We are going to understand. Now the process of filtration, when we talk about process of formation of the urine or the process,
What?
what happened? So now as we talk about the process of filtration of the blood, we are to understand what are the steps. Process of filtration of the blood, or say the process of formation of urine. Process of formation of the urine that we are going to study, and that is brought about by the filtration of the blood. What are the three steps? The three steps are: first, process known as ultrafiltration. The second step known as selective, who is talking? The second step known as selective reabsorption, and the third step known as tubular secretion. The third step known as tubular secretion. That is what we are going to study now. The process of filtration of the blood, we always say that the process of filtration of the blood is what is happening inside the kidneys. So how would the steps go? We are talking about the first part of the nephron, that is, we talk about the Malpighian corpuscle. What is Malpighian? Malpighian corpuscle. So now the first step, what is the first step of filtration of the blood? Ultrafiltration. What is the meaning of ultrafiltration? Filtration of the blood at a very high pressure. What is the definition of ultrafiltration? Filtration of the blood at a very high pressure. Where is this process happening? Here. Let us try understanding ultrafiltration. Now this is the renal artery. Is the blood flowing at a very high pressure? It is an artery, remember. Is the blood flowing at a very high pressure? Yes, it's an artery. What is the relation again? Try understanding that this is an artery. It is bringing blood from where? From the aorta. This is an artery bringing blood from the aorta. And if that is so, then obviously the pressure of the blood in this vessel is very, very high. Now, these are sets of blood capillaries, a whole network of blood capillaries that I am talking about. But to understand ultrafiltration, I may think of only one single capillary. I think of only one single capillary. So I am just erasing this to understand that what would happen in one of these capillaries only. So what would happen in one of these capillaries only? To understand that, I would say that, say suppose I talk about only one single capillary like this, but there are hundreds. So this afferent arteriole, what is this afferent arteriole? How is this afferent arteriole formed? It is a branch of the renal artery. What type of blood is it carrying? Blood is oxygenated, the blood is at high pressure. And what are these that I am talking about? I am talking about the capillaries now. These are all capillaries. How are these capillaries formed? These capillaries are formed for the division of the renal artery. What are these network of capillaries known as glomerulus? I am talking about only one single capillary right now. What are the properties of the structural features of the capillaries I mentioned? Pores, presence of pores. Two features now that you understand, the pores of the blood very high. The capillary has got pores. All of you, if you see the figure, then you will understand. Otherwise, you will not understand anything what I am speaking. So now I am saying that this inner wall of the Bowman's capsule is perforated. So I am saying here that what is the inner wall of the Bowman's capsule? One special feature of the Bowman's capsule is the inner wall has got holes, has got pores. So I would say inner wall is perforated. Very simple, just few steps to understand. The blood is flowing at a high pressure. The capillary wall has got pores. So what should happen? What is expected? The capillary wall has got pores and the blood is rushing at a high pressure. What is expected? The liquid part of the blood will come out. Same the concept for lymph we had studied. So this the same thing is happening here. A whole lot of substances are draining out. a whole lot of substances are draining out. So I would say that yes, a whole lot of substances draining out and collecting here in this depression, collecting in this inner wall, collecting in this inner depression. But at the same time, will it remain here for a long time? This fluid, will it remain here for a long time? No, it will not remain because the inner wall has got holes and it will start seeping inside. So the first thing that I'm talking about is the process known as ultrafiltration. What is happening? Blood is rushing in the capillaries at a very high pressure and hence the leaking out of a whole amount of liquid. Leaking out of a whole amount, leaking out of a large amount of the liquid. Majorly we talk about the liquid part of the blood. What is the liquid part of the blood? Plasma. So the plasma is majorly leaking out and a whole lot of substances dissolved in the plasma. So this particular process of leaking out of the blood plasma due to the high pressure of the blood and the pores present in the capillaries, what is this process known as? Ultrafiltration. How did I define the word ultrafiltration? Filtration of the blood at a very high pressure. Where is this process happening? This process is happening at the glomerulus. This process is happening at the glomerulus. So now, if you try understanding that yes, ultrafiltration is, what is the meaning of this word? Ultrafiltration means filtration of the blood at a very high pressure. Filtration of the blood at a very high pressure. So now, when we are talking about that filtration of the blood at a very high pressure, then after the filtration, what is getting formed? The liquid part of the blood all leaking out. Why? Because the capillaries always are known to have small pores in them. So now that the capillaries have got pores, the liquid part of the blood coming out, and now this fluid is formed which is getting accumulated in the small depression there. What is this fluid now known as? This fluid is now known as the glomerular filtrate. What is this fluid known as which is formed after the process of filtration? Glomerular filtrate. What is expected to be the composition of the glomerular filtrate? Yeah. It is supposed to have majorly the plasma. It is supposed to be majorly the plasma, no blood cells. But here, unlike the concept of respiration, nutrition, here we are only trying to find out what is the concentration of the nitrogenous waste because we are talking about excretion now. So what is the composition of the glomerular filtrate here? Remember, the composition of the glomerular filtrate is found to be a whole lot of water. A large amount of water is there. The glomerular filtrate has got sodium and potassium salts. The glomerular filtrate, does it have urea? Traces only. It has got traces of urea. What is the glomerular filtrate majorly made up of? What is this glomerular filtrate majorly containing? The glomerular filtrate now which seeps inside and enters into the hollow of this tube. It is entering into the hollow of this tube and running down. The glomerular filtrate is entering into the hollow of the tube and running down. Then what is the composition of the glomerular filtrate? It has got a huge amount of water. It has got a huge amount of water and it has got all the salts and the ions. But does it have the urea in large amounts? No. Urea in traces. So that easily makes us understand that this is not the only process for filtration of blood. There must be some other processes involved. Because this filtrate, now that I talk about that, what is this filtrate now known as? This is now known as the glomerular filtrate. And how much of the glomerular filtrate does our body produce? Per day, our body produces almost about 160 liters of glomerular filtrate per day. Our body produces almost about 160 liters of glomerular filtrate per day. How is it possible? We have got how much of the blood in our body? 6 liters. So that 6 liters all throughout the day, almost about 300 to 400 times goes through filtration. So if we try understanding this, that as a result of ultrafiltration, what is formed? Glomerular filtrate is formed. Where is the glomerular filtrate collected? In the depression of the Bowman's capsule. What is the composition of the glomerular filtrate? A huge amount of water, salts, ions, proteins. But at the same time, urea isn't there. Just few number of molecules only. Traces of urea only there. But it's a huge amount every time that is formed. Almost about 160, 180 liters of glomerular filtrate formed. So now that also gives us an impression that whenever we are talking about this glomerular filtrate, as it is now running down the entire tubule, some other processes also must happen. Can we afford to lose this much of water, this much of fluid from our body? We cannot. We have just 6 liters of blood and this much of water loss, meaning immediately we suffer from or immediately we collapse. But that does not happen. Even though this much of the fluid is formed in the glomerular filtrate, the body has got a method of taking back all the essential substances back and throwing out only the toxic waste substances. So we will learn how. So is this first step clear? What I am talking about? I am talking about ultrafiltration. What is the definition again? What is the definition of ultrafiltration? At high pressure. Where is this process taking place? In the glomerulus. Where is this process taking place? In the glomerulus. What is getting filtered? Ultrafiltration. What is getting filtered here? The blood. The blood is getting filtered. So how is the process happening? As the blood rushes at a very high pressure, because the capillaries, which capillaries? Glomerular capillaries, since they have got small pores all over, as the blood rushes at a very high pressure, whole lot of the liquid part of the blood starts oozing out. So whatever is remaining back here, some amount of blood is remaining there also, no? So whatever is remaining back here, if I understand, whatever amount of blood is remaining back here, is it very thick? Can I say that the blood which is remaining back here in these vessels are thick? Is it that? These capillaries will unite back to form these efferent arteriole, yes or no? As the blood rushes down and goes, the blood has lost all of its water, almost 90% of the water gone from the blood. 90% of the plasma gone from the blood. So what is remaining back in the blood now? All the blood cells, proteins, etc. So is the blood in a fluid state? No, the blood is quite thicker now. So can I say that the blood in the efferent arteriole is in a fluid state and the blood in the efferent arteriole thick? Yes. So just remember that one concept, that as the blood is entering this side and now that they are going out, the blood here is thick, the blood here is much in a fluid state, watery. The blood is watery, the blood is thick. Why? What is the reason? Because of what process? Ultrafiltration. ये आया समझ में what I told just now? Now, then only you are able to understand what is happening. Now, if I talk about a comparison, say suppose, now I talk about this blood vessels. I am talking about this blood vessels. These are the blood vessels that I draw. These are the blood vessels that I draw, parallel to this tubule. So I am now comparing. Now that the fluid, try understanding, wherever you don't understand, you'll have to ask. So I'm talking about the blood is running through this blood capillaries, blood flowing at high pressure, liquid part leaking out, the plasma part leaking out, and majority of it leaking out, forming a fluid. The fluid starts collecting in the depression. What is the composition of the fluid? Majorly the plasma. And some traces of urea also, which I mentioned straight away there. And now what will happen to this fluid? The fluid starts running inside and enters into the hollow of the tube. What is this fluid? First of all, to name it as glomerular filtrate. But as this fluid now runs down the PCT and the loop of Henle and the distal convoluted tubule, A whole lot of exchange of substances happen with the blood. Let me just talk about this part. Now, if I talk about this part, say suppose I am drawing this entire PCT. I am drawing this PCT. Say suppose now I talk about the entire PCT tube. Surrounding the PCT, what is there? Surrounding the PCT, the blood vessels. This blood vessel. I am talking about this region. What is happening in this PCT? As the glomerular filtrate comes down, what does the glomerular filtrate has? A whole lot of water. It has got sodium. It has got potassium. And what is present in the, I'm talking about the blood vessel. Now this is the blood vessel. As the blood runs down here, this is the glomerular filtrate. It has got a whole lot of water, sodium, potassium. Are you following? So as the glomerular filtrate runs down here, what am I doing? I am understanding a comparison between the blood vessel and this tubule. What is happening? From here, the water will all come down to the blood again because the blood is thicker. The blood, which blood am I talking about? The blood in the capillaries formed by the efferent arteriole. The blood in the capillaries formed by the efferent arteriole. These capillaries. The blood is thick. The water comes down. The sodium comes down. The potassium comes down. A whole lot of reabsorption happens. So this entire part I will show you this way. Try understanding. After filtration, we will just talk stepwise. I am talking about a feature something like this. This is the Bowman's capsule and the surrounding blood vessel. What am I drawing here? I am drawing that now this Bowman's capsule, the glomerular filtrate. What is this? This is the glomerular filtrate. How is glomerular filtrate formed? After the process of reabsorption. Now that the glomerular filtrate enters into the hollow, what is present here? A whole lot of water. Enough of water is there, enough of sodium, enough of potassium salt. I'm just talking about 2-3 components right now. Now, what am I drawing this? What is this? This is the blood capillary. Which blood capillary? The one which is coming away from the glomerulus. What is the next step after ultrafiltration I mentioned? Reabsorption. Why reabsorption? Because our body cannot afford to lose this much of the water. Then what does our body do? Our body will start taking back a huge amount of the water. The body will start taking back a huge amount of this water to the blood. Meaning what? From this, a lot of water goes back to the blood capillaries. From this, a lot of water goes to the blood capillaries. From this, a lot of water goes to the blood capillaries. So if I have to show you that, I am showing you this, that the water goes back to the blood again. This is the blood. The water is going back to the blood. A whole lot of water goes back to the blood. What is this process known as? This process is known as reabsorption. But I mentioned that this part, the first part of the PCT, what am I talking about? This is the PCT. Which part? This part. I'm talking about the first part of the tubule. Difficult to understand? Difficult to understand? Achha, first of all, I understand you all are not able to follow this. Huh? Who told you this?
Yes, ma'am. हो गया.
कितना हो गया?
10:46.
1 मिनट हो गया.
5 ब्रेक.
क्यों बोन्स?
2 मिनट हो गया.
Okay.
2 मिनट हो गया.
This particular, the rest of the 2 processes, again, the next class I will explain you. Ultrafiltration, ये concept समझ में आया? Is this bit understood? Those of you did not finish the diagram of the nephron, try completing this. The processes, again, the next class you have to understand. This is new for you. It will take a bit of a time to understand the relations. Tubular secretion. I'm going to explain you again the next class. Complete the diagram.
उसके बाद में
देखिए
आधे ना नोटबुक देना
ये लिख रहे हैं पढ़ के
पढ़ के 3 क्वेश्चन लिखने लास्ट
वो दो लिखना है
आधा नहीं लिखना
अबे तो इधर आके बैठ मैं पढ़ लिखना उसका ले
मुझे तो डाउट के नाम पे क्वेश्चन सर पे टिक रहा है पहले के जो 7 है
हो गया ना
वो डायरेक्ट आंसर
उनको दे दे
उसको भी नहीं लिखा
उनको दे पीछे
क्या
पूरा ही होगा इसने भी लास्ट में लिखा
उसने